Hi everybody, this is Leon. I'm a postdoc work at the Boyce Thompson Institute. So this series of video is aiming for creating online tutorial for guiding new users to work on the phenotype and the RaspiPheno app, the two computational tools we developed here to streamline the computational process of phenotypic data processing. So this video will be specifically focusing on introduction of RaspiPheno app and R-Shiny-based app for downstream data analysis. So if you want to go over some of the specific usage for phenotype, we recommend you go to the other video under the same channel. All right, so here today we get started the introduction of the Pheno app. To realize the functions, we need to download all the R files stored on our GitHub page. I'm going to paste the link below in where you can download all three .r files. These three files are global.r, server.r, and UI.r files. Three of them are necessary to launch the Shiny app. The global R files includes all the packages will be loaded for running this program. The server R are all the codes and analysis to be done. We will not touch the specific codes while running. And the UI.R is just the layout of how this R look like. So basically, we don't need to touch and modify any of these codes, but just make sure you have them ready and open them together onto your RStudio. So when we got the three files ready, we noticed that there's small green arrows indicates the wrong app. So by clicking this one, the RStudio automatically brings us back to another new page. Under this page, you can see the layout and this user interface. So it basically transformed all these codes into this graphical user interface where you could click and navigate around. So let's get started with the specific usage. Here I'm gonna use the PhenoRig, one of the infrastructure within made as examples to show how to perform the analysis. You have options to the Pheno cage. This is pretty much very similar to the PhenoRig. So to get started with, we need to upload raw files. This contains the data from the upstream analysis of the image data extraction. The first parts are Raspberry Pi derived CSV files. If you don't know what this file means, I recommend you go to the other tutorial regarding introduction of RaspiPhenotype. We also need a decoding metadata file. This one includes all the information of experiment design. If you don't know what kind of this file should include, I recommend you go to our GitHub page to refer example. So let's get started by uploading Raspberry Pi based CSV files. By clicking the browsing, it's going to bring you to a file uploader. So here we have all the images data collected under three Raspberry rigs. The Raspi M, T, and U includes images under two cameras, the camera A and B respectively. So we could select all these data as entry. So you will see all these 6 files will be uploaded, and you will be able to see them be merged here. We also want to click a specific time regarding when the analysis will be started. So we usually start the imaging at 8 a.m. in the morning. Now we further upload the decoding files. And now you can see there's a merge data bottom, which means you want to combine the specific individual plants corresponding to the metadata. So the metadata gives you an idea where the specific images belongs to certain RaspiPheno rig, which specific camera, more importantly, because there are 16 plants under the rig, and they would have a specific position. This serves as the barcode to corresponds to the RaspiPheno data, the 6 files we just uploaded and merged together. So we click the decoded data, where you can see that all the specific area at a specific time point are finally matched to individual plants for each of the camera and each of the RaspiPheno rig. And the plant.id is a unique name that is important to distinguish the individual plants across different planting rigs and cameras. All right, so after the data merging, we could quickly have a look at the visual correlation of how this data look like. By clicking the visual correlation tab here, we should usually leave it about a few seconds to upload. All right, so you can see that's the basic looking of the plant growth across a period of time. So you can distinguish your samples by given a specific variable. So here we have representing plants across 3 RaspiPheno rigs. You can switch them to cameras as well. So the biology of the experiment here we performed is to compare the stress response under control condition and water stress 1 week and 2 weeks after. So we here use the group represented by the treatment for this data visualization. If you want, you can also fast-seed variables. By clicking that, you will see that under 3 conditions, you have a clear and a more specific view to the plant growth under RaspiPM, RaspiT, and RaspiU.